Well, genomic imprinting appears to be unique to placental mammals. You find it in the marsupials, but you don't find it in the monotreme mammals, which lay eggs and don't have a placenta. And it's played a very important part in development, and since it's concerned with things like placentation, maternal commitment, and maternal care, which are major commitments of the matriline in mammals, in placental mammals, then the matriline, we must look at the matriline as having played a major role in this evolutionary process. So this is a list, by no means comprehensive, of imprinted genes which are expressed in the hypothalamus and in the placenta. And what you'll notice, two things. One is they're all maternally imprinted, and they're all paternally expressed. And a large number of them are concerned with development in terms of cell cycle arrest and apoptosis, or growth suppressors. And of course, they are co-expressed in both the hypothalamus and the placenta. And this is a slide which shows you a network of imprinted genes that is taken from a paper by Veroldt. It's a mathematical construct that is being put together to show the interactions. I'm not clear that any of these lines represent the degree of interaction. It simply tells us that there's an awful lot going on, that there's a number of downstream genes that are being affected by these key regulatory genes. And I've just put PEG3 here because PEG3, I don't know which of all of these hundreds of genes it regulates off the top of my head, but I know that it does interact with other imprinted genes, the ones that I've shown here in yellow. So there's maybe a network of imprinted genes as well. But certainly there's a very big stream of genes that are regulated by this PEG3. If we look at development, this is a LAC-Z marker of PEG3, and we see it's expressed in the developing hypothalamus, which we see here, and the developing pituitary, and it's also expressed in the placenta. This layer here is known as a spongiotrophoblast, because as you can see, it's got a lot of holes in. Those holes are the lacunae, where the mother's blood passes into, and around it you see a lot of these dark dots. These are very large cells. They're polynucleate. They're called the giant cells, and those giant cells produce hormones. And the hormones that the placenta produce has a very big impact on mother and on mother's behavior. And this is a kind of summary slide of the way these hormones act. The major hormone of pregnancy is progesterone, and that is produced by those giant cells. And progesterone acts on the brain, particularly on the hypothalamus. It acts to shut down GnRH. That makes the female infertile, and that switches off sexual behavior. It also acts to increase food intake. And food intake in the female when she's pregnant occurs way in advance of any energetic demand from the fetus. In other words, if she waited until the fetus grew to a size and then started through energetic demands to need extra food, the mother simply couldn't eat enough. So if you like, the placenta's planning the mother ahead. It's telling her to eat early so that later in pregnancy, when the demands are greater, she will be able to supply those. And likewise, there's no point the female being sexually active if she's pregnant or showing sexual behavior. It's not going to be, in reproductive terms, it's not going to have any impact. Progesterone also acts on the brain to bring about oxytocin synthesis. And this occurs through quite a long period in pregnancy. But, of course, you don't want oxytocin release. And the placenta also produces an oxytocinase for any leakage that might be there. As pregnancy progresses, so progesterone is converted to estrogen. And that conversion occurs in the fetal adrenal. And the fetal adrenal develops quite late. So the timing of it, when the adrenal is mature, then that is the time to start switching that progesterone to estrogen. And what estrogen does is it brings about an increase in oxytocin receptors. The oxytocin receptors are important for parturition, they're important for maternal behavior, and they're important for milk letdown. Another thing progesterone does actually in the brain is it's converted to pregnenolone. And pregnenolone is an anxiolytic. It reduces anxiety and calms mother down. So, the fetal placenta then exhibits forward planning by regulation of the maternal hypothalamus, and it does this by increasing maternal food intake when there's no energetic demand, by suppressing sexual behavior and fertility, by initiating nest building before offspring birth. So, how has the maternal hypothalamus evolved to plan ahead for these adaptations that respond to the fetal placenta of the next generation? So we see the fetal placenta is planning ahead, but how does the maternal hypothalamus know to respond to that? Well, one way we can look independently at what is happening in the hypothalamus and what is happening with the placenta is to mate the female, a wild-type female, with a mutant male. If it's happening in both at the same time, you've got a problem of partitioning out the two individually. But here you have a wild-type female with a normal hypothalamus, but the placentae are all carrying the mutation. We can do the converse. If we have a mutant female mated with a wild-type male, then the hypothalamus here is carrying the mutation, but all of the placentae are wild-type. And when we do this, we get a number of changes. This is, first of all, when the mutation is in the mother's hypothalamus, and what you see is there's a big change in terms of maternal care. This is a wild-type mother. You see all this white bedding that she's carried in and all the pups here underneath her. This is a mutant mother. She's not built a nest. The pups are scattered around. And we can look at a whole lot of behaviors: latency to pup retrieval, time spent to retrieve all of the pups, latency of nest building, latency of crouching over pups, and the mutant is shown here in blue, and in all cases the mutant is impaired. We've also looked to see how the hormones affect this, and if it's simply secondary to any hormones, but here we're looking at a multiparous female giving hormone replacement, and here a virgin female giving hormone replacement. PP is the postpartum female. And again, you can't overcome this by giving exogenous hormones. It's still there. And one of the consequences of this is, if we look in the hypothalamus, if we look in the paraventricular nucleus, the medial preoptic area, this is the area that is important for the oxytocin and the supraoptic that bring about parturition, bring about milk letdown. And what you can see is the number of neurons, oxytocin, these are stained for oxytocin, in the paraventricular nucleus are reduced in the PEG3 mutant, and they are reduced in the medial preoptic area. And here we're looking at oxytocinergic neurons in the hypothalamus against body weight, because these animals tend to be a bit smaller, and you see they're 25% less in the mutant. And here we're looking at the number of neurons specifically in the PVN, and again you see they're about 25% reduced. And if we look at the behavior, this has all been published, and I don't want to spend forever looking at behavior, so this is a summary of what we find. So this is PEG3 silenced in the fetal placenta. The current maternal consequences is it's priming the hypothalamus for food intake, it's priming the hypothalamus for maternal care, and it's priming the mammary gland for milk letdown. If we look at the fetal hypothalamus now, future maternal consequences, in other words, when it becomes adult, we now find that this PEG3 silence in this has an impact on regulating food intake, regulating maternal care, and regulating milk letdown. So you've got functions that are co-adaptive. They're in the same domain, as it were. They're very similar functions when you selectively put the mutation in the hypothalamus or selectively put the mutation in the placenta, which is what makes me think that they're co-adapted. And one way we can look at this, here's the fetal placenta. signaling, talking to the maternal hypothalamus. Here's the fetus developing its hypothalamus. Co-expressed genes, co-expressed imprinted genes, are in these two structures at the same time, and this fetus hypothalamus becomes an adult hypothalamus. We can add a bit of solidity to this. Here we see the imprinted gene in the placenta and in the fetus. Now that developing hypothalamus of the fetus becomes an adult hypothalamus, and we move to the next generation where this imprinted gene is affecting the placenta and the feedback. So what you've got here is you've got a different kind of template for selection pressures, because depending on how this developing placenta talks to this adult hypothalamus, when this fetal hypothalamus is developing by the same genes, same key regulatory genes, this will serve as a template to ensure that this hypothalamus will be functioning in the same way in the next generation. And if that's true, if what I've said is true, then we might expect that if we looked at co-expressed genes in the placenta and in the brain, then what we would find is that some are co-expressed, which we see here in red, some are unique to the brain, hypothalamus that is, and some are unique to the placenta. And about E11.12 of development, you can see that most of them are in the placenta, and only 9% of all genes that are changing their expression over developmental day 11 to 12 are in both placenta and hypothalamus. But by E12 to 13, PEG3 is having a very big impact on these co-expressed genes. We see that now 40% of those genes are impaired with the mutation, and only 16% in the placenta and 37% in the brain. But this tallies with what I was saying before, this big increase in terms of co-expressed genes at this stage is probably due to some of these placental genes now going to be co-expressed with the developing hypothalamus. And if we look at this co-expression over time and look at the PEG3 mutation, this is what I've just shown you here at 11 to 12, and now what we see is with the mutation, the number of co-expressed genes actually increases. But E12 to 13, what we see is there's no change. They're pretty much the same, and there's no change in any of these. So this, for PEG3, is a critical time in development, 11-12, for the co-adaptive development of the two structures. And we can also see what happens if we look at these genes in a different way. We're looking here at genes which are induced and genes which are suppressed and genes which are unchanged. And we're looking at PEG3 and also PEG1. I'll tell you why we're looking at PEG1 in a minute, because it complements what is happening in PEG3. It has many of the same behavioral impacts, and it is too co-expressed in brain and placenta, and it complements it. But what you see is with this early stage, you find that 74% of new genes are expressed, which is why you get a large percentage at this time period in development. And what we find is a very small number of genes are suppressed. But all genes which are changing expression are affected. But by E12.5, it's only about 50% are not affected and 50% are. PEG1, on the other hand, has very little effect. 70% are unchanged at this time point. But by the time we get to E12.3, we see that it's going up. 25% that are co-expressed are now being changed. So, this begs the question of why maternal imprinting and paternal expression. One thing that is crucial with viviparity and placentation and internal development is you've got to get development correct. You can't afford any errors. Any errors represents a major loss of investment for the mother, not only in terms of her offspring, but others that are in the litter, and also for the mother herself. And so gene dosage, critical control over gene dosage, occurs by genomic imprinting because you have monoallelic gene expression, always the same allele. And the other thing is you need stability. Now, that is counterintuitive. If I'm saying these genes are very important in evolutionary terms and have had a major role in evolution, you would be expecting them to change. But in fact, you don't. They don't change. There's papers that have come out which have looked at imprinted genes across many mammalian species, and they are remarkably conserved. There are very few point mutations, and there are very few translocations, virtually none. In fact, it led the paper, which was actually looking for differences, but they had to come to the conclusion that these imprinted genes are undergoing purifying selection. So that will certainly render them stable. And that is, of course, these genes are also, the ones I'm talking about now, are paternally expressed. And that's counterintuitive, because you get many more mutations being brought about through the patriline due to the vast number of cell divisions you get. So whatever is happening through the patriline, certainly DNA repair is mandatory post-fertilization. Well, one of the things we find is that post-fertilization, you get maternal regulation of paternal reprogramming in the zygote. That is active demethylation, post-fertilization in the zygote, and this occurs in the context of TET3 and mismatch long patch base excision repair. And here, I'm showing you what part, if you like. Here we're talking about male genome reprogramming, but the matriline is playing a major role. You've got active demethylation, TET3 UG mismatch long patch BAR repair by UNG, and cytosine deamination. So this occurs during the nuclear replication phase. And of course, the sperm, they're tightly packaged with protamines. And they don't undergo transcription until the second, third nuclear replication. And they're not carrying very much cytoplasm, so they're not carrying many factors through into this generation. So one of the things that you find is that the protamines have to be removed by maternal factors, and a lot of the histones, because protamines replaced histones in the sperm, a lot of the histones have to be replaced, and many of these histones are maternal histones. It's also the case that there are maternal zygotic factors. So this is a gonadal-specific expression gene, and this action demethylates the paternal alleles. This TRIM28, and TRIM28 regulates epigenetic stability. PGC7 or Stella protects the maternal imprints from demethylation. If you've got active methylation occurring in the zygote, then clearly you want to protect the maternal alleles. And this maternal factor, Stella, and same with this zinc finger protein, they protect maternal imprints. And CDX2, which is also a maternal factor in these early stages, it is produced from the male genome, it's biallelically produced later. This determines the trophectoderm lineage and its specific differentiation. And the trophectoderm is going to form the placenta. So there's a biased maternal input to this process. And we know that this is true because in a lot of the stem cell biology, if you enucleate the oocyte, the egg, you can put a progenitor cell in there and it will reprogram it. So the oocyte has this carrying capacity for reprogram. So again, why maternal imprinting, paternal expression? And I've told you about stability now, DNA repair post-fertilization in the zygote, and also the control over gene dosage through imprinted genes. They provide for stability. But of course, you do want variability as well in evolution. And I think the matriline uses spermatogenesis as a driver for duplication and variance in downstream genes. And if we look across mammals, there are three categories of genes which seem to have expanded exponentially. These are the pregnancy-specific glycoproteins, the CCAMs, the cell adhesion molecules, and the prolactins. In the marsupial, which only has 6 imprinted genes, you see these are very low. In the mouth, they're increasing exponentially. And in the human, they're also, secams and prolactins are also increasing quite considerably. So these are very important downstream genes, and the question is, have this expansion, what impact have they had on this, this action of imprinting and its co-expression and its impact on maternalism? So this is looking at the effects of PEG3 on these downstream genes. So these are PEG3 target genes. This is the same time point, embryonic 12 to 13, and this is in the hypothalamus and placenta where they're co-expressed. And these are the prolactins here. These are the pregnancy-specific glycoproteins, and these are the secams. And those in yellow are suppressed. Those in white are unchanged by PEG3 mutation, and those that are in blue are desynchronized. Most of those desynchronizations are up here. These are either suppressed or unchanged or upregulated. So you're getting some, I beg your pardon, you're getting some which are desynchronized in the, especially in these placental specific glycoproteins in the hypothalamus and the placenta. Now what is happening is these two, we don't know very much about any of these. We know a lot about the prolactins. We know the prolactins are cytokines. We know the prolactins also induce progesterone. And we know that the prolactins act in the brain to stimulate the oligodendrocyte precursors. The pregnancy-specific glycoproteins, we know that these 23 and 21 are important for binding to the CDK9 receptor on the NK cells, so what we call the innate immune system. The innate immune system is important for bringing about invasion. It's not the standard rejection. It promotes invasion and promotes angiogenesis. And the other PSGs, 17 and 18, are very important for angiogenesis. Again, we don't know what these are doing in the brain, but if they stimulate angiogenesis in the placenta, chances are they're going to be doing the same thing at the same time in the hypothalamus. And the CEAs, again, these are cell adhesion molecules. We don't know very much about these. We know that proteoglycans, and this is a glycoprotein, we know that these are important in neural nets. We don't know whether they're important in the syncytium of the placenta, which is an important structure. The other thing that we find with these placental glycoproteins is that they too are important in angiogenesis. And if those two that I was telling you about, I think it was 17 and 18, if those two are impaired, or rather they are impaired, correlationally that is, when you look at preeclampsia, when you look at small for age gestation babies, and they also can be responsible for abortion. So those placental specific glycoproteins are important. So, the matrilineal contribution to reproductive success has depended on the co-adaptation and co-adaptive evolution of brain and placenta. And this co-adaptation underpins a common evolutionary goal. It coordinates mother-infant physiological homeostasis, it ensures maternal energetic investment, and it provides for focused maternal care. So the evolutionary outcome of this linked co-adaptation is that offspring which have extracted good, in inverted commas, maternal nurturing will be well provisioned for and genetically predisposed towards reproductive success in the next generation. Of course, these effects, mother-infant physiological homeostasis, maternal energetic investment occur or are equally important for male offspring as they are for female offspring. But male offspring don't provide for focused maternal care. So why is that? Why are there all of the advantages going to the male, but the male is not involved in any of the engagement of commitment to the offspring? Well, we know that males have a different reproductive strategy in terms of their success. Their success comes from mating with as many females as possible. Females are restricted by estrus as to the number of matings they can have and the number of pregnancies they can have. So a mouse, for example, which has a feral life of about 30 days, can only produce around about 10 weeks, I beg your pardon, can only produce around about 3 litters. And so every litter counts, everything is important. But the male can mate with some 300 females in this same time period if he's up to it. And so what is happening in the male? Well, we know that you get masculinization in the patriline. We know you get testosterone acting on the hypothalamus and testosterone acting on the placenta. PEG3 is expressed in both of these tissues. PEG3 is also expressed in the fetal vomeronasal organ. And you'll know from a lot of the works by Catherine Dulac and others that the vomeronasal organ is crucially important for male sexual behavior. Without the vomeronasal organ, the male will mate anything, virtually. He cannot recognize a female, he cannot certainly recognize an estrus female, and his reproductive success goes way down, and he virtually stops mating. So testosterone is important for acting on the hypothalamus and the vomeronasal organ at this time to masculinize it. Now, that testosterone during development comes from what is called fetal Leydig cells. These are different from the postpubertal Leydig cells because they don't need LH, they don't need the pituitary to stimulate them to produce testosterone. And after birth, they stop producing testosterone and they undergo apoptosis. Whether or not it's the same stem cell population that produces the adult Leydig cells is not known. People think it is, but nobody's actually shown that yet. But of course, these fetal Leydig cells are dependent on SOX9, and SOX9 is dependent on SRY, the sex-determining gene. And SRY is a gene which is unique to placental mammals. You don't find it in the monotreme mammals. You only find it in the placental mammals. And the textbooks will tell you this is a new gene, an important new gene. But in fact, it's not a new gene. They've found out recently that it's a hybrid gene. And it's a hybrid of two old genes, SOX3, which is important for developing the hypothalamus, and diGeorge critical region 8, which codes for Droshap complex and is important in placental development. So that could be a coincidence, but I think that one of the things that is going to be important in the development of these fetal Leydig cells and the production of the testosterone is timing. And in order to get that timing right, you need to know in the developmental process of those Leydig cells where they are with respect to the placenta and where they are with respect to the hypothalamus. So that may be potentially a way in which that timing has been brought about through SRY. Well, that stimulation of the vomeronasal organ by testosterone brings about the masculinization. You get sex differences at the first relay. This is where the receptors are. You get sex differences at the first relay, the second relay in the bed nucleus of the stria, and at the medial amygdala, and in the medial preoptic area where this convergence occurs. And if you expose males to estrus female urine tells the males about their reproductive condition. If you expose them to that, one of the things you find is that in the exposed versus the control, at all of these relays, you find that exposing the male brings about an increase in CFOS activity. CFOS is an indicator of neurons which are being activated. So more neurons are being activated all along these pathways to the medial preoptic area, which is important in the male for regulating sexual behavior, and of course in the female for regulating maternal behavior. So this attractiveness, and which will affect male sexual behavior, is also influenced by these by the testosterone and the masculinization. But also, if we look at the PEG3 mutation, what we find is that if we look at investigations, this is the wild type, and this is estrus urine versus male urine, and it's crucially important to distinguish between them. They show a lot of investigation of estrus urine, but with the PEG3, you find, and this is a sexually experienced animal, you find that that has gone down significantly. They still inspect it more, but it has gone down somewhat. And likewise, if we look at mount latency, here we're looking at the wild type, and here we're looking at the PEG3 sexually experienced female. And we're looking at all of these behaviors: mount latency, intromission latency, sexual interaction frequency, and the percent of males that are achieving ejaculation. And what you can see is that compared with the wild types, the PEG3 mutation is severely impaired. So we're finding then that the same gene, which is co-expressed in the hypothalamus and placenta, the placental effects on that hypothalamus in the male are avoided by masculinization. And that masculinization is also, the PEG3 impairment is seen in the male through that masculinization. So if we look at neural activity, this is in the accessory olfactory bulb, this is looking at the CFOS, and we see in all of these relays neural activity is up when you're exposed to female urine. If we look at the adult, we find that PEG3 knockout, and you're exposed to the urine, that it is not increased. It is it goes down, if anything. There's no effect compared with the wild type control. And also what we find is at the same time in all of these relays, you get an increase in caspase 3. And caspase 3 is concerned with apoptosis, and so what you're finding is there's a decrease in the neurons of this pathway, which is probably bringing about the ability to as it were, make use of this information and produce the behavior. So, in conclusion then, viviparity has been a primary driver of mammalian evolution and reproductive success. Hypothalamic evolution was highly dependent on coadaptation with the fetal placenta. And genomic imprinting, under matrilineal control, the genome, the imprints are methylated in the matriline, is a primary regulator of maternalism and male sexual behavior. SRY evolved to counteract maternalism in males, and imprinted gene which regulates maternal care also regulates male sexual behavior via the vomeronasal pheromone pathway. But I think it's important, the emphasis, well, as I said at the beginning, two genomes interacting in one individual. In actual fact, it's three genomes if we take it to its ultimate determination, because not only do you have the mother and the mother's fertilized egg, but you also have the eggs that are developing, the oocytes that are developing in the developing ovary in the mother. So you have these three generations developing in this one individual, two in the mother and one in the fetus of the mother. And in the literature, you hear, when they're talking about reprogramming, when they're talking about reprogramming of the genome, you usually see it says the male genome is reprogrammed first, immediately after fertilization in the zygote, and the female genome, the female imprints, are reprogrammed later in development, round about E11.5, 10.5 to 12.5. And that's true from a developmental point of view. But what you have to remember is those imprints which are being reprogrammed around about 11, day 10 to 12, they are for the next generation. So that by the time that egg comes to be fertilized in the next generation, the maternal imprints and the maternal genome is already reprogrammed. And the paternal genome then has to protect against reprogramming from the male reprogramming process. And at the same time, there are factors that are there in the oocyte that allow that to happen. So, finally then, this work is being done by students of mine, James Curley, and Will Swanee and Kevin Broad. Naoki was a postdoc from the States. Francis was a postdoc from the States as well. Azim made the PEG3 and the PEG1 knockouts. And I actually did take part in the work. I did do a lot of the actual experimental work myself. So thank you very much indeed. I'll answer questions.